Every expectant mother knows that what she eats impacts her baby. And now research shows that is also true for our cows. Maternal consumption of ReaShure during late gestation had a positive effect on the in utero calf, setting her up for better health and potentially even higher milk production once she joins the milking string. Learn more at balchemanh.com/launch and launch your herd for life.
A question that was sent in to get us going here. Any evidence that enhanced feeding of methyl donors to late gestation cows under heat stress provides any protection against heat stress implications on the calves?
Amanda, you want to take that one?
I was trying to figure out
You're trying to figure your mic out.
in vivo data, not that much, and not with choline that I'm aware of. Methyl donors in general in vitro have been shown to improve, I'm thinking in mammary epithelial cells, milk protein genes and proliferation in general, but specifically with choline, I'm not aware of.
So the Florida studies that I shared, they were, as you said, there's 200 days of heat stress in Florida. So those studies were, I think the calvings in those studies, they started maybe in October, and they calved through maybe April to May in both of those studies. So they would have been exposed to some heat stress, but they were not calving in the middle of the summer in Florida.
Any questions from the audience?
Can I ask a follow-up question just while we're waiting? So, Clay, I'm curious about, what about places where calving is synchronized? Because that would provide a lot more sort of temporal data for a larger group.
Yeah.
Anything happening?
So we're running some studies with, you need larger herds with tighter calving intervals. So there is some work of that. There is some of that work going on right now. It's a good question.
Just curious.
Yeah.
Clay.
Yes.
Right here. The first question that was asked, that was one that was on my mind. All these transition studies have pretty much just been 21 days, and my question was, is there value in going through the whole dry period or not? I guess we don't really have that kind of data at this point in time. Correct?
Great question. Stay tuned.
Okay. My other question, this is kind of unrelated to the calf stuff you went. I'm just curious, any differences in those choline treatments on the cows relative to actual colostrum volume production?
So that we saw it in the first study. So that first study that was published in 2018, there was a numerical increase in colostrum yield in the choline supplemented dams. There was a significant improvement in colostrum quality in that first study. So IgG yield would have been much higher in that first study from the choline supplemented dams. We did not see that in the second study at Florida. Now, what I would say in the second study, the colostrum quality was much higher than even in the control animals in the second study. But we didn't see it in both studies. We did see it the first time.
Were they done in different seasons of the year?
No, it's a good question. The difference actually was what my understanding is the difference, they were milked at different times post-calving between the two studies. So I will say we've gotten reports of that from the field for years. That was one of the reasons why we wanted to look at this. We had heard that before.
Yeah, so we've heard a lot about sort of these negative stresses that will cause an epigenetic change. Now that can persist over maybe even multiple generations. I'm wondering what is the mechanism that sort of changes this back? And is there any way to sort of reverse these negative impacts with some sort of positive intervention for the animal or something like that?
That might be a question for Kevin, because I was kind of wondering if my mom and my grandma didn't have enough choline in their diet, am I stuck?
Yeah, I would. Can you guys hear me?
Yes.
I would say that is still a really active area of investigation. There's a little bit of work that's been done in like the xenoestrogen exposure literature with bisphenol A and prenatal programming, where later caloric restriction could reverse a lot of the epigenetic reprogramming. That's obviously extremely disparate. But yeah, I don't think we have any idea. And I think, the choline's effect on cognition and things like that, there are definitely changes in gene expression and likely, histone and DNA methylation changes. But whether they're actually downstream of choline's use as just a methyl donor because of increased cholinergic signaling or something related to phosphatidylcholine, it's really unclear at this point in time. So we don't know the exact causal thing. We know it's downstream of choline, but it could be downstream of any of its major metabolites, and how we target that is still not clear. Maybe one day we'll have CRISPR that can really keenly go to individual parts of the genome and clip off methyl groups or add methyl groups, but we're not quite there yet with in vivo editing of epigenotype.
I was just going to echo that we know that those changes are triggered by stressors. We know that they are stable and they can, be evident 2 years and even 40 years. I don't remember who was showing that. I think you both. 40 years after, you still see those differences, but we don't know how we can revert them yet. So I think that's, as Kevin said, an active area of investigation. And I think we all agree that we know very little about, we definitely have more questions than answers, but there are enzymes that will revert at least DNA methylation. What triggers that? I don't think we know.
So the only example that I can think of is there are some examples of in tumor biology where there's some misregulation and tumors grow because of an aberrant methylation pattern, and then a sort of a very aggressive chemotherapy is to treat the patient with a chemotherapy that essentially erases DNA methylation, and then it has to start over again. And when that happens, those tumors are able to be eliminated by the immune system. They become targeted and they go away. And so there is a sort of a brute force option, but that wouldn't be practical in animal agriculture.
We'll take a question that was sent in virtually here. The 63 days of life to study in the chicken is attractive. What are the limitations for extrapolating to humans?
Limitations to extrapolating to humans. If we think about, what are the limitations? I will say the bird is an understudied model organism when it comes to human, as a clinical model. The 63 days is roughly equivalent to that first 1,000 days of human development. And we were talking at the break that if you think about sexual maturity, those chickens don't become sexually mature until they're about 16 to 20 weeks of age. And so if you think about a human, that would be about 4 times 1,000 days, or maybe 12 or 13 years old. So there is that similarity there. I think there is opportunity if during that first 63 days the animal is plastic, if we can manipulate things that would have long-term impacts. We should be working in those generations higher on that population pyramid instead of focusing our work at the bottom, because if we can make changes at the top, we can have one animal that could influence 4 billion animals, or 4 million animals, sorry, at the bottom of the pyramid. So I think there is potential to look beyond that shortened period in those animals that are destined to be the progenitors of those subsequent layers of the structure.
Another question from the audience?
Thanks, Luke. I've got one for Kevin. Kevin, in dairy cattle, we supplement choline the last 21 days of gestation. From your research in humans, what's the optimal time to supplement with choline?
Yeah, so in our most recent trial, we went back pretty early between gestational week 12 to 16, which I believe is the earliest anybody has gone in humans and supplemented. And the primary outcome of that trial was looking at omega-3 fatty acid availability. So we had some reason to believe that the habitual choline intakes exacerbate the methyl donor stress of pregnancy, and that supplementation might alleviate that and allow you to basically improve the DHA economy is kind of how I like to think of it. So it was a very targeted nutrient stress or nutrient interaction that we were looking at, alleviating the stress on the choline end of things to see if we can optimize the DHA side of things. But we have ongoing cognitive outcomes. Not that DHA isn't important as an outcome on its own, but I think people care about most the hard endpoints of cognition. So we're very eager to see how the cognitive phenotyping from this study compares to the previous study, and see if that longer period of choline supplementation might be beneficial. Because in the prior study, the control arm got higher doses of choline, but we also intervened at the time where choline stress becomes greatest. Now we think we've gone earlier and sort of improved one carbon nutrient status going prior to and then going into that nutrient stressor. So I guess it sort of depends on what outcome you're looking at. And we don't have much data. This is one of the longest trials that has supplemented with a choline salt for this long. There was one trial that supplemented with phosphatidylcholine for almost as long as ours that did not find improvements in cognitive outcomes. But for anybody who's worked with PC in humans, you know that it takes like 5 to 7 pills a day of someone taking on top of prenatals and DHA and whatnot. So it can be, I think some folks question how adherent people are, but we need a lot more literature. I think practically speaking, women start prenatal vitamins like soon after figuring out that they're pregnant, just as more of an insurance policy, and that's likely when a recommendation to consume a choline supplement would occur as well. So with humans, it's always practicality as well as physiology.
I've got one here, Luke.
So this kind of piggybacks on what Corin was talking about as far as length of supplementation. What about dosage during heat stress times? Have you altered dosage to see its effect, increase, decrease? And also with different breeds, say Jersey versus Holstein.
Good questions. Don't know the answer necessarily to either of those because we haven't actually run those studies. So very good question. We really don't know the breed effect. I mean, pretty much everything's been done. All the controlled research we've done has been in Holsteins for the most part. There have been a few other breeds in some of those studies. So can't really make a different recommendation at this point based on breed. The heat stress question is a good one that we'll take a deeper look at that. Right. Yeah. We've done some dose response work. A lot of it's been done in a restricted fed dry cow model that so they're dry cows where you feed ad libitum first and then you restrict feed intake. And very rapidly they will develop fatty liver. It's a great model for that. So you increase liver triglycerides very, very rapidly. We've done dose response work there, and there definitely are benefits to higher doses in that model, but we've not done much of that work actually, from a production standpoint in transition cows.
Got a question that came in via the internet. Is it possible to overmethylate? Can one turn off or on too many genes?
I don't think we know the answer to that question. Theoretically, I think it would be possible to overmethylate, and you would it's like hitting the mute button on your television. Biology usually finds a way to make it through, so I'm not sure how you would. I'm not sure how you would overmethylate. I guess that's what I'm trying to. Any other ideas?
Yeah, I think just because an animal has the capability of methylating something doesn't mean that it will.
Necessarily will,
and
yeah.
Feeding methyl donors gives them the capability of doing something as opposed to forcing them to do it.
Yeah, I think.
You want that there, but not necessarily ready to methylate. I guess the cell will dictate, but the methyl donor should be there.
Right. So what we were saying is the methyl donors are the reservoir to adapt to the stimulus that's coming from wherever it's coming. And if they're not there, then
So you don't want it to be limited for sure.
Right. You don't want it to be limited. So if they are limited, then the animal can't respond adequately. I'm not sure. It's possible.
So, Chad, I have a question for you. So what determines if a gene will be upregulated or downregulated?
I have no idea. It's just the cell and its environment, right? And what it needs, the protein it needs at that specific point in time.
Another question from the audience?
This question is for Dr. Laporta. In terms of the financial implications of heat stress, do you think that those costs are going to continue to increase as we keep seeing record high temperatures across the United States, especially in the Midwest and Western U.S.?
Yeah, definitely. And we have done some simulations to actually test that. And, in this type of analysis, you can be overestimating some things due to the assumptions you make when you run these models. But we try to be quite conservative, but doing those simulations. And yes, it's definitely possible because, we're doing everything we can to fight climate change and, individual efforts and collective efforts, but the trends are here. So, more and more cows are going to be exposed, unfortunately, and people. So, we definitely need to take measures before they actually get heat stress, so we don't have to do things after. So, preventative measures. But yeah.
And it might not be just ambient temperature. It could be, storm-related power outage, which causes, it's different.
So how long do these dry cows have to be cooled to get the beneficial effects?
How long?
Yeah, how long during the dry period?
So we have targeted 46 days. The typical dry period length has been 60 days, but we've shown that 46, it's enough time to, from a mammary selfish perspective, right, to regenerate, to get rid of those senescent cells and give time to regeneration and calostrogenesis before parturition. So we have target 46 days. But in our last study, we did a little longer because it was in a commercial farm. So we did, and in primiparous heifers, we did 55 days. And obviously, you see the same effects. But 46 days is what we target. But we also ran an experiment where we did just the first 3 weeks of the dry period, or the entire dry period, or just the last 3 weeks, and you also see detriments in the calf and in the cow. So not necessarily the entire dry period. So at any time for at least 2-3 weeks, they will see the negative repercussions. So.
Got another question that kind of follows up to that one there from outside the meeting here. The impact of heat stress on mammary gland morphology was profound, but what about other organs, liver, lungs, immune system?
That's a great question. So obviously our center is the mammary gland, but we have been looking at liver as well. Skin. The skin is greatly impacted. The sweat glands, these calves are born with less sweat glands. The adrenal gland is something that we're looking into as well. The ovaries that I just showed, these calves, these heifers have lower AMH concentrations throughout the pre-weaning period. We don't really know what that means, but like we're looking into that. They have smaller follicles and stuff like that. So yeah, it's obviously heat stress impacts the whole animal, not just the mammary gland. I show a few examples, but yeah, it's a holistic impact for sure. Yeah. So we're looking into more of other organs as well, but metabolism in general, it's impacted. So.
Great. Another question from the audience.
Do you have any thoughts on episodic heat stress as opposed to the chronic type that you would see in Florida?
Yeah, I haven't done any work on that yet because I was 5 years in Florida, so that's what you do, right? Chronic heat stress. But yeah, others have shown very clearly that episodic heat stress also impacts the animal. I'm not aware of studies showing long-term effects, so typically the animals recuperate depending on the intensity and the duration, obviously, but the lactating cow will recuperate. But what we're showing here is the chronic heat stress having long-term effects. I'm not aware of episodic heat stress during the dry period, how that might impact the calf or the cow. I don't know that. But that's a great question. Stay tuned.
And this is a follow-up maybe question that's similar to that. Are subsequent exposure to stressors such as heat stress creating compounding or additive impacts on the same genes, either up or down regulated in subsequent generations?
So if I understand, is exposure after, like the additive effect of in utero? So we ran a study where calves were exposed in utero and then postnatally all across. So that was about 110 days. All along, or we switched them. Once they were born, we put them in a cool environment or in a heat stress environment. And what we see is that the effects are cumulative. So the the calves that underwent heat stress in utero and then postnatally, they do okay until like they reach the hottest time of the day and they have higher respiration rates, higher rectal temperatures, and their lagging growth. It's also evident. So yes, it is cumulative. Yes.
Another question from the audience. If not, we have one for Dr. Klatt. As you listen to the questions related to the animal work, what suggestions from the human medicine research are we missing and should be considering going forward?
Oh gosh. I have a few thoughts on that, I guess. One that I think even the human medicine world forgets is dietary betaine. We tend to forget about it. We're just not thinking about it all that much. Sorry, one second. Little puppies in the background. We tend to forget about betaine or not quantify it, and that's certainly, choline is being oxidized to betaine, and there might be, as we think about epigenetics and methyl donors, something to consider. There's also, in the sort of lab animal world, there's been more interest in phosphatidylcholine as a potential ligand for nuclear receptors. So we're thinking about choline stress here and potentially increasing phosphatidylcholine levels. And we've heard it's a surfactant, we've heard it's a lipotrope, but it's also able to bind specific nuclear receptors that are relevant for the cow, and probably other species as well that might be mediating its transcriptional effects. So those are a few things that come up off the top of my head, but there are so many things that we could think about in just dose-response relationships and key developmental windows and whatnot that we're all still teasing apart that I think everyone appreciates the importance of, but we all kind of do single-dose studies versus a control, and really dose-response relationships are needed, and you need an additional arm at least to start to get at that.
Thank you.
Oh, that was the last thing I should say too is microbiome. And so leaching, in the cow, it's a whole different world. We have foregut fermentation, but in lab animals and in humans, we think about leaching of choline and formation of trimethylamine and how that's affecting the total choline dose. And so in different GI conditions, there's concerns that you might need increased choline because there's increased conversion of choline to TMA. And that might be relevant in various situations and in pathologies in the animal agricultural world.
Great, thank you. Another question from the audience?
I have one for Clay. Is the choline reassure, is that rumen protected?
Yes.
Okay.
Yeah. It needs to be. Yeah, there's very little that gets through the rumen if it's not rumen protected.
At Balchem Animal Nutrition and Health, we strive every day to deliver results you can see in your animal's productivity and your bottom line. From a smooth transition into the milking string for your fresh cows, to a happy welcome home from your furry friend. From a strong start in your poultry flock, to consistent weight gains for your finishing hogs. We expect to earn your business and your trust with our people, our products, and our science. Our people have an intense passion for your animals and your success. You can count on us for honest, candid advice and practical solutions for your toughest challenges. As the global leader in choline production, chelation, and encapsulation technology, we take our obligation to you and to the environment seriously. Our products are backed by the most extensive and thorough research portfolio, while our business is committed to advancing environmental sustainability and animal welfare. In the end, it all comes down to results. Balchem delivers real results you can count on, results that exceed your expectations, and results that bring true value to your bottom line. Leading the charge to meet the nutritional needs of ruminants, monogastrics, and companion animals, Balchem offers a growing portfolio of nutritional products and a dedication to innovation and industry sustainability. Balchem is here to solve today and shape tomorrow.